Hello, BISC130. This is the beginning of recorded lecture 3-4. Still in this genetics chapter. So everything we talked about last time in the last recorded lecture was Mendelian genetics, about this, Gregor Mendel, and we talked about Mendelian inheritance patterns, all this stuff, dominant, recessive, all those fun things. Now let's talk about something different, non-Mendelian inheritance. So it is true that there are many genes and traits that follow everything we talked about last time, but there are exceptions to this. So I'm double underlining this because there are going to be several things underneath this, but yeah, for the next while, we'll be talking about things that sort of break some of the rules that we defined last time. One of these is something called polygenic inheritance. So what we learned last time was that one gene controls one trait. There was one gene that controlled the phenotype of whether the pea plants were purple or white. But in polygenic inheritance, the phenotype is not the result of one gene, but the phenotype is the result of two or more genes. A bunch of different genes, all contributing to the same phenotype. Now, when the phenotype was only controlled by one gene, we only had two options. The flowers were either purple or white. But when we have a bunch of different genes and they're all contributing to a single phenotype, we get what's called continuous variation within the phenotype. The best easily observable example of this is a lot of human traits, including skin tone. There aren't just two options for hair color or skin color or eye color in humans. There's a whole spectrum of pale to dark and everything in between. It's because this phenotype is not controlled by a single gene. It's controlled by a bunch of genes. So continuous variation refers to the phenotype having a spectrum of different phenotypes, of different possibilities. The key terms define continuous variation as an inheritance pattern in which a trait or phenotype shows a range of values. And again, just to call out one particular example, skin tone in humans, easy example of this. This is a result of having multiple genes contribute to a single phenotype. Okay, that's all I had for polygenic inheritance. Again, we're going to be going through these. There are several examples of non-Mendelian inheritance. Another one is something called incomplete dominance. So, in incomplete dominance, the heterozygote individuals are somewhere intermediate in appearance between the two homozygotes. And again, this is totally not what we talked about last time. What we talked about with classic Mendelian genetics, the heterozygote has the dominant phenotype. It's not light purple. It's not a mixture of purple and white. In Mendelian inheritance, the heterozygous individual has the dominant phenotype. But if we're talking about this incomplete dominance, the heterozygotes are going to be a blend. They're going to be somewhere in between. This is really not very common. These are snapdragon flowers that do display this incomplete dominance. And yeah, you can see here's the red allele, here's the white allele, the heterozygote is pink. It's in between red and white. So not that complicated to understand, but it is a rarity in nature for sure. Incomplete dominance, heterozygotes are somewhere in between the two homozygotes. Okay, next we have something called codominance, which I'll read the key terms definition now, but it's exactly what it sounds like, codominance. Codominance is the complete and simultaneous expression of both alleles in a heterozygote. So this is not blending the two alleles together. Codominance is both alleles are on and fully active and making their thing at the same time. This is hard to wrap your brain around. We will see an example of this in just a second because I'm going to do a 2-for-1. So I will show you an example of codominance, but I will also show you an example of our next thing. So just hold on to this for a second. The idea of two different phenotypes being expressed at the same time. Hold on to that idea. There will be an example coming up as we talk about multiple alleles. So this is another just example of non-Mendelian inheritance. You could have more than two. So yeah, in the first part of this chapter, we had a purple allele and a white allele, just those two options, but alleles are simply different versions of a gene. There's nothing stopping them from being 3 or 4 or more than that. Multiple alleles refers to a situation where there are just more than two. There's a great example of this in humans, our blood types. So the ABO blood types in humans are, as the name implies, 3 possible alleles here. The A allele, the B allele, and the O allele, so 3 alleles here in this system. O is recessive, classically so, just as we talked about recessive, in Mendelian genetics. A is dominant, B is dominant, and they are codominant with one another. So yeah, I told you this would be, an example of codominance and an example of multiple alleles. So what does this actually look like? Well, to understand this, we have to look at what these blood types actually mean. So the blood types refer to a specific protein present on the surface of our red blood cells. If you have an A allele, your red blood cells will have the A version of this protein on their surface. If you have a B allele, your red blood cells will have the B version of this protein on their surface. Here's the codominance. If you have an A allele and a B allele, if you've got AB type blood, it's pretty rare in our population, but if you've got AB type blood, there's the codominance. You are making the A version of this protein and the B version of this protein on the surface of your red blood cell. So that's how we can have both alleles expressing themselves at the same time. That's the codominance. And the O allele is defective. If you have an O allele, it doesn't make any version of this particular protein on the surface of the red blood cells. And so if we want to make a little table for this, again, with 3 alleles, there are more than just 2 possible genotypes. These are all the possible genotypes and phenotypes. It's easy to fill out, instead of having to just memorize this table. But yeah, if you've got an A allele and another A allele, of course, you're going to have A-type blood. If you've got an A allele and an O allele, remember O is recessive. So if you've got one gene saying make the A version and another gene saying I don't know what to make, you're going to make A version. Just like we saw with purple and white, a defective allele here doesn't do anything, but it doesn't get in the way of any other alleles that may be there. So A allele and O allele results in A-type blood. A B allele and another B allele, obviously you're going to have B-type blood. If you've got a B allele and an O allele, just like we said here, O is recessive, you're going to have B-type blood. If you've got an O allele and another O allele, you're going to have this recessive phenotype. You're going to have O-type blood. And then here's the codominance. If you've got an A allele and a B allele, you're going to have AB-type blood. So interestingly, if you have O-type blood, by the way, if you don't know your blood type, I know the Life Share is a blood donation place that has trucks that come by campus often enough. If they're ever on campus and you see those and you're, health-wise, you're able to donate blood, that's always a good thing to do. They always need blood for people in hospitals. But as a fringe benefit to you, if you donate blood with them, you get a little account and a little card and everything, and they'll tell you important information about your health, your blood pressure and cholesterol levels, and your blood type. So if you don't know your blood type, you can donate blood and you'll know your blood type. If you've got O-type blood, that's your phenotype. You actually know your genotype. You know exactly which genes you have because there's only one way to have O-type blood. Same thing with AB. If you've got AB-type blood, you know exactly which alleles you have for this gene. However, if you've got A-type blood, you don't necessarily know. You could be AA or you could be AO. If you've got B-type blood, it could be BB or it could be BO. So yeah, interesting to look at. Very applicable to humans and stuff. And yeah, donate blood if you're able to. One last thing about blood, just because in class someone usually asks about this. They ask why this matters. The short version is your immune system freaks out against proteins it is not familiar with. So if you've got A-type blood, your immune system is used to seeing the A version. But if you've got A-type blood and you are transfused with B versions, your body is going to attack that, and it's going to be bad. Yeah, and if you've got B-type blood, you're used to the B protein. But if you're given either of these types, your body's going to have a negative reaction against it. O-type blood, your body's going to freak out against any of this stuff, but you'll be fine with receiving other O-type blood. This also means O-type blood, because it doesn't have any copies of this protein, any version of this protein, anyone can accept O-type blood. So if you have O-type blood and you've got it, it is even more important for you to donate blood if you are able to, when you are able to, because yeah, it's sort of the universal donor, it's sometimes called, because yeah, A can accept O-type blood, there's nothing to offend them here. B can accept O-type blood, there's nothing that's going to trigger an immune response. AB can accept O-type blood, there's nothing here that they'll react against. Then of course O can receive O-type blood as well. So anyway, I'm not writing any of that down. There's just usually several people who are curious about why this matters. It has to do with immune reactions and reacting against things that you're not familiar with proteins you're not familiar with. Okay. What's our next example? So again, still under this umbrella of non-Mendelian inheritance, let's talk about something called recessive lethal. This refers to an allele that is lethal, deadly, but only when you have two of it. If you're heterozygous for this lethal allele, it's not lethal. There may be a phenotype associated with it. There may not be a phenotype associated with it, but it's not lethal in its heterozygous form. This allele is lethal in its homozygous form. Example, more cats. Here's a Manx cat. So this is a cat that does not have a tail or has an incredibly short tail. It's due to this defective stub tail gene. If you follow this up here, the healthy allele is the lowercase, the stub tail gene is the uppercase, the capital. Again, S, not a letter I would choose, especially if you're doing handwriting here. S is very confusable, but again, stub tail S, that's why they picked S. So yeah, here's the heterozygous individual with only one copy of this recessive lethal allele, and yeah, it's alive. It has a phenotype, it has a stub tail, but it's not dead. And yeah, this is kind of showing a Punnett square. So one stub parent and another stub parent, one heterozygote crossed with another heterozygote. These are our possible offspring: normal tail, stub tail, stub tail. Here's the lethality. So recessive lethal, it is lethal when you have two copies of it. So here's stubtail allele and a second stubtail allele. This is catastrophic to growth and development. This is going to be lethal to the embryo early on. Dead. But again, in its heterozygous form, not lethal. Lethal in homozygous. So, yep. Lethal in homozygous, in heterozygous, may have a phenotype or may not have a phenotype. And yep, the example here is the Manx cat. Embryonic lethal in the homozygote. Heterozygote has simply a stub tail. All right. Next, we've got something called X-linked traits. X-linked traits are where the gene for this trait is located on the X chromosome. Now, at this point, we need to do a bit of a sidebar. So you could note this in your notes however you want to. I'm doing this here. We have to go on a bit of a side tangent so we understand what an X chromosome actually is. Then we'll come back to X-linked traits. So most mammals, including ourselves, use this XY system for biological sex determination. So we only have one sex chromosome. The X and the Y are two different versions of that sex chromosome. So these are kind of like alleles. Alleles are two different versions of a given gene. X and Y are two different versions of an entire chromosome. So we have two different versions of this chromosome. And yep, don't sweat the details here, but yeah, here's an X chromosome. It's rather large compared to everything else. It has a lot of important genes. Here's the Y chromosome. It is much, much, much smaller. We'll see why in just a second. But yeah, these are two different versions of the sex chromosome. And we saw this, coming back to this for like, what, the third time? Eukaryotic chromosomes, yeah. 2 copies of chromosome 1, 2 copies of chromosome 2, 2 copies of chromosome 3. If we skip down, there's the 23rd chromosome, the sex chromosome. It looks like this individual has an X and another X. But yeah, we've got 2 copies of the sex chromosome. Side note to the sidebar. So these are the sex chromosomes. Everything else is called an autosome. So all other chromosomes, other than the sex chromosomes, are called autosomes. They're not directly involved in sexual development. So what is the difference between these two? So we got 2 different versions. What do they do? Well, the X chromosome is involved in sex development, but it's also just required for life. There's a ton of genes among these, 1,000 genes on the X chromosome that you just need to live. We cannot live without an X chromosome. So the X chromosome has many genes not related to sex development that are just straight up required to be alive. And the other genes within the X chromosome direct development toward female sex. It just has instructions for, during development, build a uterus, build ovaries, like go through this process of building female sex. The Y chromosome does not have genes that are required for life. Again, it is much smaller than the X chromosome. It doesn't have anything that we strictly need. So yeah, you can survive without a Y chromosome. About half the people on the planet are surviving just fine without a Y chromosome. Y chromosome is not required for life. And what it does is it in some ways just alters the X instructions to lead to male sex. Oh, don't build ovaries, build testes instead. So it's sort of taking for granted that anyone with a Y chromosome also has an X chromosome, because everyone has an X chromosome because it's required for life. That's why the Y chromosome is much smaller. It's just taking the instructions of the X and changing them a little bit, leading to male sex. So again, we are diploid. We have 2 copies of each chromosome. So our possibilities here are an X chromosome and another X chromosome. The instructions here are going to lead to the development of female sex. If you have an X chromosome and a Y chromosome, the instructions here, especially on this Y, are going to change things, lead to the development of male sex. If you're looking at the possible combinations here, yeah, what about YY? Well, this is not normally possible with the way eggs and sperm work. And even if it were possible and you were to create a cell that had a Y chromosome and another Y chromosome, it would not be viable. Remember, the X chromosome has genes that are required for life. So any cell that had 2 Y chromosomes and no X chromosome at all would not be able to survive. That would be lethal to development. One other thing in this sidebar is this is not the only way to do things. So, in humans and most mammals, and oddly enough in fruit flies as well, there is this XY system. Females have 2 of the same thing, males have 1 of each. But in birds, for example, it's the exact opposite. In birds, males have 2 of the same sex chromosome, and it's females that have 2 different ones. When we get to invertebrates, it gets even wackier, with just differing numbers of chromosomes, and things like grasshoppers and honeybees are really weird. The males are just straight up haploid. There's no dedicated sex chromosomes, just every single chromosome the males only have 1 of, and the females are diploid and have 2 of. So don't memorize this table, but it is interesting to look at. My point here, in other animals, other than mammals, other than humans, sex can be determined through different mechanisms. And even not on this list, not even listed on this table, There are many reptiles that determine sex not using genetics at all. There are many crocodilians that determine male-female sex based on the temperature of the eggs as they're developing. So yeah, there are other ways to do this. The XY is just how mammals do this. So end of sidebar. All of this was trying to explain what X-linked trait is supposed to mean. So an X-linked trait is a trait where this gene, for this phenotype, for this trait, is located on the X chromosome. This matters because XY individuals only have one X chromosome. For every other gene, we are diploid. We have two copies. It matters whether it's dominant or recessive. But if you're XY, if you only have one X chromosome, there is no backup copy. It doesn't matter whether it's dominant or recessive. You get what you get. There's only one of these that you have. So if this is a disease associated with the X chromosome, XY individuals are going to be more likely to have this because they only have one copy of that gene. They don't have a backup. Red-green color blindness is an example of this. I can't verify this, but I guess if you're red-green colorblind, these two images should look exactly the same. But red-green colorblindness, again, nothing to do with sex development at all. The genes for this are on the X chromosome. So if you're XY and you've got one defective copy, you're going to be red-green colorblind. It is possible for XX individuals to be red-green colorblind, but they would have to have two defective copies. It's just more difficult for that to happen. So yeah, this is what I mean. XY individuals are more likely to have these X-linked traits. Duchenne muscular dystrophy and SCID X1, it's a severe immune system disorder. Those are also X-linked traits and are much more commonly seen in XY individuals. Okay. One more. One more example of non-Mendelian inheritance. And this is kind of a big one, but I think it's a pretty interesting one. It is something called epistasis. Now, it's highlighted, it's in the key terms, but I actually don't want to start by reading the key terms definition. I want to, walk through this process, and then we'll read the key terms definition. So, an example of epistasis is coat color in mice. So let's look at these mices. We got three options. We've got a agouti. Okay, it's brown. I don't know why mice people call this agouti, but it's agouti, also known as brown. We've got black. We've got white, also known as albino. Now, there are two genes involved in determining what the coat color of the mouse is. Aha, you may say, that's polygenic inheritance. Well, it kind of is polygenic inheritance. This is, two or more genes contributing to the phenotype, but as we will see, epistasis and what's going on with these mice is not just two or more genes. It's a little more complicated than that. So one of these genes controls what the color of the pigment is. So the agouti, or brown, the agouti allele is dominant. And hey, capital A, it works this time, but yeah, the letter we choose is usually associated with a dominant phenotype. So the capital A allele, the agouti or brown is dominant. The black, again, not B, these are the same gene, they're two different versions of the gene. The black allele, lowercase a, is recessive. So yeah, brown is dominant, black is recessive. That is one gene controlling what the color of the pigment is going to be. There is a separate gene, a second gene, that controls whether the pigment is actually made or not. Normal production, capital C, I'm not sure why it's C, whatever, is dominant, but no pigment being produced, which results in an albino phenotype, that is recessive, that is lowercase c. The reason why this is not just polygenic is because among these two genes, one controlling the color of the pigment and one controlling whether the pigment is produced or not, this gene overrides this gene. The pigment production gene is sort of stronger than the pigment color gene. It doesn't matter what the pigment color gene is saying, this is going to sort of take precedent over it. Let's look at how this can play out. So this is essentially a massive Punnett square tracking these two genes. Don't memorize, the exact numbers or percentages here. Let's just look at all the possible combinations of mice. So let's focus on the A gene first. Capital A, capital A. It's brown. It's agouti. Duh. Heterozygous, capital A, lowercase a. Remember, agouti is dominant and black is recessive. So heterozygotes are going to be brown. Capital A, brown. Capital A, lowercase a, brown. Capital A, lowercase a, brown. I mean, all these things are brown. To look at some black mice, lowercase a, black. It's the recessive phenotype. The only way to have the recessive phenotype is to be homozygous recessive. There's another homozygous recessive. There's another homozygous recessive. All right, let's start paying attention to the C alleles. Remember, capital C means normal pigment production. So capital C, that means, okay, other gene, go ahead, do your thing. In this case, brown. Capital C, okay, other gene, do your thing. In this case, brown. Here's capital C, lowercase c. Again, normal pigment production is dominant, no pigment production or albino is recessive. So being heterozygous for this pigment production gene means, yep, go ahead, first gene, do your thing. In this case, resulting in agouti. And we see this here in the black mice. Capital C, yep, go ahead and have black pigment. Capital C, lowercase c, yep, go ahead, have black pigment. But let's look at these. Lowercase c. That means no pigment. This is supposed to be an agouti mouse, according to this gene. But because this pigment production gene overrides the effects of this first gene, it's going to be albino. No pigment production, doesn't matter what the A gene is saying. Here's another agouti mouse that is now albino because of lowercase c. Here's a black mouse, it's supposed to be making black pigment, but that black pigment is overridden by the lowercase c. So epistasis is not just two different alleles or multiple alleles, it is multiple genes where one is stronger than the other. So let me read the key terms definition of epistasis here. The key terms define this as an antagonistic interaction between genes such that one gene masks or interferes with the expression of another. We see this here with the pigment production gene, the C gene, masking the effects or interfering with the effects of this first gene, the black or agouti pigment. So again, we had it written down before. In summary, the lowercase c mice are white regardless of what the agouti black alleles say. This gene overrides the other gene. Okay, that brings us to the end of non-Mendelian inheritance and to the end of genetics, but we're actually not done with this recorded lecture with the last few minutes. I want to skip over and briefly get started on the next chapter, chapter 14, DNA structure and function. To understand the topics in this chapter, we're going to need some background about DNA. And actually, we've kind of done this already, but remember, my class is not supposed to be cumulative, so even though I talked about this sort of stuff on exam number 1, I'm not going to test you over it again unless I bring it up again. So everything I'm bringing up again here is fair game for this exam as well. We really need a refresher about a lot of this stuff in order to understand this chapter on how DNA works. So let's start with this statement. DNA is made of nucleotides. DNA is a polymer of nucleotides, a bunch of these individual structures that are put together. Phosphate, 5 carbon sugar here, nitrogenous base, not uracil. That's RNA only. DNA can have adenine, guanine, thymine, or cytosine. So, to summarize, nucleotides consist of a 5 carbon sugar, a negatively charged phosphate group, and a nitrogenous base, which could be adenine A, guanine G, cytosine C, or thymine T. No uracil, that's an RNA thing. The 5' phosphate of one nucleotide is connected to the 3' OH of the next in a phosphodiester bond. This should look familiar to you, and yep, there is the 5' phosphate connected to the 3' OH. 5' phosphate connected to 3' OH. Here's a 5' phosphate, and if we were to continue building this strand of DNA, there would be a 3' OH of a nucleotide that would come in here and become attached to this 5' phosphate. Again, these terms, 5' and 3', going to be very important as we go through this chapter. The 5' end has the phosphate, the 3' end has the OH. They're always connected to one another in this way. Now, we don't just have one strand, we've got two. There are 2 strands of DNA that form a double helix structure. The integrity of this double helix is held together by hydrogen bonds between these bases. So 2 strands of DNA form a double helix, held together by H bonds between the bases in a very specific way. Thymine partners up with adenine, guanine partners up with cytosine. If you hadn't drilled this into your brain already in the earlier chapter, here it is again. These are the rules for base complementarity. C pairs with G, A pairs with T. That is how they optimize the strength of their hydrogen bonds. Any strand of DNA is going to have a nucleotide opposite it, according to these rules. C's are partnered up with G's, A's are partnered up with T's. Now, all of this so far should be familiar. It should be review from several chapters ago. There is one new thing that I want to introduce now that will be very important when we talk about the rest of the stuff in this chapter. And it has once again to do with the 5' and 3' stuff. So however long this strand is, doesn't matter whether it's 3 nucleotides long or 3 million nucleotides long. If it's a line of nucleotides, there's always going to be one end with a free phosphate, not attached to any partner. And the other end is going to be a free 3' OH, not attached to any phosphate partner. We can see these things labeled on lots of these figures here. So circle this. Yeah, here is the 3' end of this long string. This is the 5' end of this long string. And yep, here's a more detailed view of it. But yeah, here's the 5' phosphate that is free. It's the very end of this DNA molecule. Here's the 3' end down here, the OH that doesn't have any partner. Yeah, doesn't matter how long it is. Any strand of DNA is going to have a 5' end and a 3' end. Now, the other important thing, is that in a DNA double helix, these 2 strands of DNA do not go in the same direction. They're pointed in opposite directions. So notice here, the strand on the left, it just happens to be drawn so the 3' is at the top and the 5' is at the bottom. The other strand is going to be opposite. Its 5' is at the top, its 3' is at the bottom. We see this over here in the more detailed one as well. The one on the left is running 5' to 3'. The strand on the right is running 5' from the bottom to the 3' at the top. 2 strands of DNA in the double helix run. Again, the technical term is not opposite. The technical term is anti-parallel. They run anti-parallel to one another. They're going 5' to 3', but in opposite directions. We can actually kind of draw this ourselves if you want to draw a piece of DNA. I am not a great artist, which is why I rely on, great figures like this. But for some things, I can draw this myself. Even if I were writing on paper, I can draw 2 lines. There's my DNA. If we wanted to label this, let's say this top strand has 5' on the left and 3' on the right. Because the 2 strands are always anti-parallel to one another, if we were to label the bottom strand here, the bottom strand would be labeled like this: 3' on the left, 5' on the right. Again, get comfortable with 5', 3', and get comfortable with the idea of them running in opposite directions. We're going to see a lot of 5', 3' stuff as we get deeper into this chapter. Speaking of which, we'll get deeper into this chapter in the next recorded lecture. I just briefly wanted to get started on DNA structure function in this recorded lecture so that we could really start things in earnest in the next one. So this is the end of recorded lecture 3-4.